All right, so if we've got lots and lots of hydrogen ions up here in that intermembrane space, now they can go through another protein channel right here. I'm going to draw this hydrogen ion in. It's going to go through the protein channel and to this side, H+, back to the matrix. Now that is not going to need to be active transport because we're going from high concentration up here to low concentration down here. So that is actually going to be facilitated transport because it's diffusing from high to low concentration, but it still needs a protein channel to get those ions through. So to get the ions into the intermembrane space, we need active transport. To get them into the matrix, we need facilitated transport. Now, hopefully the point you're at now, you're asking yourself, why would your cells pump all those hydrogen ions and use energy to move them into the intermembrane space only to let them diffuse back through again through that protein channel? So that hopefully is where you're at right now, and that's a good place to be. It means you're understanding what's going on. So I'm going to erase what is going on with this drawing here. Hopefully you remember most of this. And go through the answer now of why are these hydrogens moving? What does this have to do with making energy? So we're going to do a few more labels. This little oval right here, which is attached to the protein channel, which is right here, is an enzyme. And the enzyme is called ATP synthase. So SYNTHASE. And if you remember from enzymes, what they actually do is speed up reactions. And based on the name of this enzyme, you can figure out what it does. Synthesis of something means you're making something. And what are we making? We're making ATP. So what it does, this enzyme, is it speeds up the reaction of taking ADP and adding a phosphate to make ATP. That's our reaction. So ATP synthase makes this reaction happen. There's ADP and phosphate sitting here in the matrix, and the ATP makes it happen, makes it actually go quick enough. Now, why do we need hydrogen ions? That's the question I left you with before. When the hydrogen ions diffuse through this protein channel and interact with this enzyme right here, this ATP synthase, they actually activate the enzyme. They change the shape of the enzyme so that it can perform this reaction better. What I like to think of it as is kind of like a water wheel on an old-fashioned like grain mill or something, or not grain, yeah, grain mill. So, you know, they had those buildings that used to be on rivers, and they'd have the water wheel that would sit right on the river. And as the water flowed over the water wheel, here goes the water, it turned the water wheel and made the grinding stone inside the mill actually grind up whatever it was they were grinding. Usually it was wheat for flour. So you can think of this action as our hydrogen ions. So our hydrogen ions are going through the channel and by the enzyme. Our enzyme is our water mill, okay, our water wheel. And by doing that, they kind of make the enzyme work. They make it do its function. They turn it, if you want to think about it that way. If there are no hydrogen ions flowing through, the enzyme doesn't work. Okay, it doesn't spin, if that's how you want to think about it. So the whole reason that we go through glycolysis and the citric acid cycle is to make these hydrogen ions that activate ATP synthase. And then ATP synthase does the work of making this reaction happen quickly, so we make lots of ATP. In fact, with this enzyme activated, per glucose we make 32 ATP. That's what this little star is supposed to represent, the 32 ATP that are made. So quite a bit more than was made in glycolysis in the citric acid cycle. All right, so the last thing I want to point out to you is these little guys right over here, because they are important, and I need to talk to you about how this works. So I said that we pump hydrogen ions from this matrix area into the inner membrane space using energy. Okay, where does that energy come from? Well, these guys, these hydrogen ions, when they break off, they're not alone. What else comes off, because we're breaking bonds, are some electrons. And I'm going to represent that with an E with a little dash on top of it. So not only do we lose hydrogen ions from the NADH and the FADH2, but we also lose some electrons. These ovals here that I'm highlighting in pink represent what we call electron carriers. They're special molecules embedded in the membrane that are good at transferring electrons. So the electrons that are released from NADH and FADH2 first start with this original electron carrier and then are passed down from one electron carrier to the other, kind of like a game of hot potato, if you ever played that when you were a kid. Pass the ball around the circle and you didn't want to get caught with it, right? So they pass on the electron from carrier to carrier to carrier, and basically it's giving off energy with each time that it gets passed down. So here, this first carrier, the electron level's energy is pretty high. As it gets passed down, it gets to a lower energy level. Here it's at an even lower energy level. And what's happening to that energy? That's the energy that's used to pump these hydrogen ions into that inner membrane space. Okay? That is called the electron, I'm going to abbreviate here, transport chain, sometimes ETC. Okay? So in order to get the energy to pump those hydrogen ions into the inner membrane space, we need to use the electron transport chain. The oxidative phosphorylation is happening back here, as I said, when we take the ADP and add the phosphate to make a TP through the help of this enzyme that speeds up the whole reaction. All right, now the last thing we haven't talked about is this oxidative part. So when that final electron is done being passed down from carrier to carrier, it has to get sent somewhere where it's basically used up, so that we don't have just random electrons floating around in your cells. And if you remember, one of the elements that we studied back when we talked about electronegativity that's really good at grabbing electrons, remember we said it was our electron bully, was oxygen. So here's why you need oxygen for cellular respiration, for your body really. Okay? What happens is this last electron then gets grabbed by oxygen, and along with the hydrogen that's still in the matrix, makes water. So, thinking back to our original chemical equation for respiration, we said glucose, which is used in glycolysis, plus oxygen, which is our final electron acceptor, right? It takes that leftover electron, yields ATP, which is our energy here, plus water, here's the water that's given off as a byproduct, plus carbon dioxide. Remember, carbon dioxide was made back in the citric acid cycle when you were breaking down those carbon molecules from 6 to 5 to 4 to 4 to 4. So all of those products and reactants are now accounted for in these three processes.